Hello, my name is Rachel Fields and my supervisors are Professor Derek Moot and Associate Professor Graham Burrell. I'm a PhD student at Lincoln University and my thesis was Causes of Elevated Coumestrol in Lucerne and Mitigation of the Subsequent Risk to Ewe Reproductive Performance. So Lucerne is a high quality feed. It has high protein as it's a nitrogen fixer, and it's also drought tolerant with a long taproot and high water use efficiency. It's becoming increasingly popular in New Zealand grazing systems. However, some farmers are reluctant to grow Lucerne as it can contain phyto-oestrogens, which may reduce fertility of ewes. So phyto-oestrogens are plant-produced steroids that bind to the estrogen receptor. Some can cause infertility, such as formininetin in older cultivars of subterranean clover, but the main phytoestrogen in Lucerne is the Coumestrol. As you can see in the bottom right, those hydroxyl groups, the OHs, are similar to the placement in the estradiol molecule on the left, and this helps them bind with estrogen receptors. So Coumestrol consumed in Lucerne by ewes during the mating period can lower ovulation rate, which reduces fecundity, or multiple births. So you have fewer twins and more single lambs. The Coumestrol is not always high in Lucerne. It ranges from 0 to about 600 milligrams per kilogram of dry matter, with levels over 25 milligrams per kilogram sufficient to reduce ewe ovulation rates. Coumestrol is predominantly produced in response to fungal pathogens, but it has been reported or thought to be produced due to flowering or water stress. Flowering, however, often they were following one stand through the growing season, and then as it was flowering and a bit older, the levels were getting higher. And the water stress seems to be purely anecdotal, that some farmers are avoiding the Lucerne if it is wilting. So for my thesis I had two aims. The first was to identify Lucerne crops that may be at risk of high Coumestrol. The second was to develop management strategies to decrease the risk of depressed ewe ovulation rates. So first I isolated factors that can increase or minimise Coumestrol accumulation. I'm going to talk about 4 of the factors that I was looking at. The first was development stage and cutting frequency, and so again this was following a stand through the growing season, but in my experiment we used different cutting frequencies so that the plants were simultaneously at different stages, so that we could simultaneously sample, say, a vegetative, a flowering and a seed set crop. In the Cultivar experiment we looked at 5 cultivars: Kaituna, Stamina 5, Stamina 6 GT, and Rhino and Runner, and this was done at Ashley Dean Sheep Farm through an autumn season. We also looked at fungicide treatment. So some of the older papers have shown that weekly applications of fungicide can be effective at reducing the fungal pathogens and controlling Coumestrol accumulation. However, weekly applications is really not economic for especially dryland sheep farmers, and so we wanted to know if using less frequent application at critical times would be more useful. And finally we looked at water stress. This was a glasshouse experiment where we compared well-watered plants with water stressed, and then plants that were so water stressed that the foliage had died. So from the development stage, where we looked at plants at simultaneously different stages, we can see that there was no correlation between Coumestrol and the development stage. For example, if we look at the yellow triangles, on the 11th of March we had plants that were at a development stage of 2, which was a vegetative plant about 15 to 30 centimetres tall, plants at stage 6, which was flowering, and then plants between stage 8 and 10, which was seed set. And these were all relatively low. So some of them were above that red line though, where we are starting to look at risk to the animals. In contrast, if we look at the blue squares, we again had plants that were at that sort of 2 level, and then we had, which was that vegetative, up to the flowering at stage 6, and the ones at stage 8 and 10. And so there really was no correlation between that development stage, and we seem to just see sudden spikes in all the plants regardless of their age. So When we compared the cultivars, we found that cultivar did not affect the Coumestrol content. So we can see that all of the plants had a similar level of Coumestrol, and the ones that were the highest sort of changed up a bit. And if we look at the first half, they were sort of below that 25 milligrams per kilogram where it's looking like it could be a problem. But then in May they were all above that 25 milligrams per kilogram where it could be a problem. So the modern Lucerne cultivars were still capable of producing at-risk Coumestrol levels. Further studies are required to identify whether some cultivars currently on the market are more suitable than the ones that we tested. And finally, we looked at the water stress. So Lucerne's an important dryland crop, which means it is often exposed to conditions of water deficit, and when it's really dry it may be the only plant available, so we don't want to avoid grazing it if it's the only green material. So in the cultivar experiment we did see that the plants on the left, this was after quite a summer drought and the plants weren't growing very well. You can see there was no canopy closure and the plants were quite stunted, but they had relatively lower. They had 22 milligrams per kilogram of dry matter relative to later in the season after there'd been plenty of rain, and the levels had more than doubled to 52 milligrams per kilogram dry matter. This seemed to suggest that there wasn't an effect of water stress. So we looked at it in the glasshouse and we stressed the plants until they had wilted, and then we took some samples and then we continued stressing them until they had senesced. You can see in the bottom right what they looked like after that time. And all of the Coumestrol contents were well below that 25 milligrams per kilogram dry matter, indicating that it is perfectly safe to be grazing water-stressed plants as long as there is no fungal damage also present. We then tested the fungicide, so we had 500 mils per hectare of Protec, which contained 500 grams per litre of carbendazim, applied plus or minus to the Lucerne crops of Stamina 5 cultivar. There was no effect of the carbendazim treatments on either the Coumestrol or the severity of fungal symptoms. So these blue arrows are the dates where we applied the fungicide, and you can see that following the applications in January and in April, the Coumestrol levels increased in both the control and the fungicide plants. So fungal damage is the main factor that Coumestrol responds to, and we saw this in our field experiments as well. However, it's not a strong predictor of Coumestrol, and it's very subjective. So if a couple of people went out in the field, they might both get different results when they score the plants on the scale of 1 to 5, where 1 is undamaged and then 5 is severely damaged. And then it's hard to say where the boundary is between the negligible and problematic level of fungal damage. So we wanted to develop a predictive tool to estimate the Coumestrol using weather data and other agronomic data. We looked at factors such as rainfall, temperature, relative humidity, which was the days with average relative humidity above 70, 80, 90 or 95%. We looked at soil moisture deficit, radiation, and then the agronomic data, regrowth age, height, yield, development stage and month. We found that days above 95% relative humidity was the best predictor of Coumestrol content. There was quite a good relationship with an R-squared of 0.74, and you can see that as there are more days with an average relative humidity of 95%, the Coumestrol levels also increase. We then made a decision tree which showed, if we look in the left hand side of the tree, with a rainfall of less than 61 millimetres and days above relative humidity of less than 5.5 days, the Coumestrol levels were predicted to be a safe average of about 7.9 milligrams per kilogram. But when you have more relative humidity, the average would be 32 milligrams per kilogram of dry matter. And the same thing when you had more rainfall. For example, if you had over 131 millimetres of rainfall and over 17 days of 95% relative humidity, you could expect an average Coumestrol content of 163 milligrams per kilogram dry matter. So the third objective was to quantify ewe responses to elevated coumestrol. So what happened in the ewe when the coumestrol was high? We looked at ewe morphological development and then recovery from coumestrol. The ewe morphological development was an observational study at Creedmoor Farm, which was 15 kilometres southwest of Oamaru. At weaning, the farmers allocated the ewe lambs by weight, with the heavy lambs on grass and the light lambs on Lucerne. So this was a management decision to increase the live weight of the smaller animals. So the farmers hoped that by putting their lighter animals on the Lucerne, they would gain weight and catch up with the heavier lambs that they put on the grass. However, in about January, the farmers noticed that the animals on Lucerne were starting to grow udders. And so we came down in mid-March and we took measurements of teat length, teat width and the mammary gland diameters. The farmers then moved the Lucerne-fed lambs onto grass, as they were concerned that the phyto-oestrogens in the Lucerne would affect their lambing rates. The lambs were then mated 5 weeks later. So when we went and visited in mid-March, 19 of the 36 Lucerne-fed lambs had protruding udders, and they also had wider teats. In contrast, none of the grass-fed lambs had udders. We then assessed the effect of diet and mating weight on the scanning rate. The diet did not have an effect, so there was no effect of the lambs having previously grazed on the Lucerne and then been removed for 5 weeks versus the ones that were on grass throughout. However, there was an effect of mating weight, and this would be expected with higher mating rates having greater scanning and lambing rates. So this was a graph that shows the expected fetuses per ewe versus the mating weight based on the Creedmoor data. So you can see that when the ewe lambs had a mating weight of 40 kilograms, they were expected to have less than 1 fetus per ewe. When this increased up to sort of 55 kilograms, they were expected to have about 1.8 fetuses per ewe, and then once they were getting to 65, they were predicted to have about 2.25 fetuses per ewe. So ewes, as I mentioned before, they are often put onto high quality feed to increase their mating weight, and so for this purpose the low weight lambs at weaning were put onto Lucerne, and then removal from the Lucerne 5 weeks prior to mating resulted in no effective scanning, and there was only a live weight effect. So the ewes in that experiment recovered after 5 weeks, so we wanted to know if shorter terms would be adequate. So this next experiment tested the length of time required for ewes to recover from the effects of coumestrol on ovulation rate. In this experiment we had 2 tooth ewes with 15 per treatment, and then 4 grazing treatments, and the ewes were all blocked by live weight and then break fed with a back fence. The coumestrol in the Lucerne throughout the experiment had an average of 29 milligrams per kilogram of dry matter. There was no effective date on the coumestrol, so it didn't increase or decrease across the time. And so we had the 4 grazing treatments, which were 6 weeks on Lucerne prior to ovulation, 3 weeks on Lucerne, then 3 weeks on grass, 5 weeks on Lucerne, then 1 week on grass, or 6 weeks on grass. And so all the animals were synchronized with seeders so that they would ovulate at the same time. And the ovulation was measured by laparoscopy to count the corpora lutea. So the highest ovulation was in the ewes that were on grass for 6 or 3 weeks prior to ovulation, 2.07 and 2.0 corpora lutea per ewe on average. There was a lower number of corpora lutea in ewes that were on Lucerne, with 1.47 corpora lutea per ewe, and an intermediate and not significantly different amount when the ewes were removed for 1 week, with 1.79 corpora lutea per ewe. 2 weeks off the Lucerne was sufficient for recovery from a moderately oestrogenic crop. So we did the same sort of analysis that we did with the Creedmoor, where we predicted the expected ovulation rate based on the days on grass, and so as the days on grass increased, the expected ovulation rate also increased, within that first 2 weeks. So taking the ewes off for 0 days, so that they were on grass throughout, they had an expected ovulation rate of about 1.5. This increased quite quickly with 7 days on grass to about 1.7, and then up close to, or maybe 1.9 at 14 days on grass, before this levelled off with further time on the grass not giving any additional benefits. So from my study, we identified Lucerne crops that may be at risk of high Coumestrol. To do this, look for fungal damage, check ewe lambs for udder development, and also compare them to ewe lambs on grass rather than Lucerne, so that it's easier to see if there is a difference. And consider that Lucerne may be a risk if there are more than 6 days over 95% average relative humidity, or over 60 millimetres of rain during the regrowth period. We don't need to worry about development stage, so you can graze when it's flowering, and we don't need to worry about water stress, so we can graze when the lucerne is wilted or dry. The second aim was to develop management strategies to decrease the risk of depressed ewe ovulation rate. So the removal of ewes 2 weeks before mating was sufficient for recovery from moderately estrogenic lucerne. Current cultivars can still produce at-risk levels, so we can't advise on cultivar strategies, and we also can't advise on fungicide application at this stage. I would like to acknowledge my excellent supervisors, Professor Derek Moot and Associate Professor Graham Burrell, for all of their help and advice with this project. I would also like to thank Beef and Lamb New Zealand, Struthers Trust and Mr. Roland Steed for financial support. I was also given a lot of advice from Drs. Richard Sedcole, Anna Mills, Alan Gash and N. Harvey. And I received a lot of technical support from Martin Welby and Jenny Zhao. And I'd especially like to thank Sharon and Julian Price of Creedmoor Super Sheep for getting in contact with us when they noticed the udder development in their animals. And finally, I'd like to thank the fantastic Field Research Centre Nursery and JML technicians at Lincoln University.